You either are a human being or you are not. And when a human egg is fertilized by a human sperm, it forms a human being at the single cell stage. The potential is how much development you can undergo. But the species designation is still the same, whether you're one cell or whether you're 100 trillion cells.
Hello, I'm Dr. Christina Francis, the CEO at AAPLOG. Before we get into this week's episode, we wanted to go over some breaking news that occurred right after we had recorded it. We recorded this episode on Thursday, October 16th, 2025. Shortly after we recorded it, the Trump administration announced a new plan to lower the cost of fertility medications and expand insurance coverage options for IVF and other fertility treatments. AAPLOG is heartened by this renewed interest in addressing infertility and helping build strong families across our country, as we will discuss in this episode of the podcast. As OBGYNs and women's healthcare professionals, we know well the heartbreak experienced by families who struggle with infertility. However, it's very important to recognize that IVF does not treat the causes of infertility, but rather attempts to circumvent them, which in some circumstances can lead to the exploitation and loss of human life. As referenced in our guidance on the ethical treatment of human embryos, the document that we're discussing on today's podcast, we encourage first-line therapies like restorative reproductive medicine that not only address and treat root causes of infertility, but also avoid the intentional loss of human life. We also support the conscience rights of any medical professional or employer that does not desire to participate in interventions that could cause loss of life. Our hope is that elected officials, policymakers, and the medical professional community will continue to pursue knowledge and expertise in approaches that both respect life and improve the health of all of our patients. Let's jump into today's episode. Welcome to Caring for Both, a curbside consult series by the American Association of Pro-Life Obstetricians and Gynecologists, or AAPLOG. On this podcast, we bring on experts to provide insights to support you as a medical professional in your life-affirming practice and career. I'm your host for today, Dr. Christina Francis. I'm a board-certified OBGYN as well as the CEO at AAPLOG. And I'm joined today by my dear friend, Dr. David Prentice. David is a PhD, and he's also president and founding board member of the Science Alliance for Life and Technology, or SALT, which is a great acronym. He has almost 50 years experience as a scientific researcher, professor, academic leader, and policy advisor. He earned his PhD in biochemistry from the University of Kansas and did postdoctoral work in cell biology. He was instrumental in establishing Kansas's Midwest Stem Cell Therapy Center, as well as Stem Cell Research Facts, an educational website about adult stem cells. He has provided scientific lectures, policy briefings, and legislative testimonies at the federal level, as well as in 40 states and 21 countries. So he's been very, very busy. Dr. Prentice, thank you so much for joining us today.
Great to be with you, Christina. Thank you.
Absolutely. So today you and I are going to be discussing AAPLOG's committee opinion that came out towards the end of last year, I think, on the ethical treatment of human embryos. Of course, this is a topic that you are very familiar with, and in fact, you were instrumental in helping author this document as well. I think considering how we treat the youngest and most vulnerable amongst us is a crucial component of bioethics and really informs how we view all of our patients when we think about how do we view human beings at their very beginning and at their most vulnerable stage. So I think this was such an important document for us to get out, and I'm really excited about our conversation today. All right, so first off, we like to start off by just getting to know our guests a little bit better. So can you just tell us a little bit more about yourself and specifically what drew you to the study of cellular biology and looking at human embryology as well?
Well, I grew up on a farm in Kansas and was just fascinated with nature, with life, and so on. A couple of quick examples. A friend had a calf die on their farm, so we did an autopsy. And we kept the heart in a glass of formaldehyde until I went off to college and my mom threw it away. She couldn't stand it anymore. But as I went through undergraduate, I actually started out looking more at ecological research, and I did a summer doing research in upstate New York. But one of the focuses was to look at the plankton and the zooplankton in the water, the tiny little creatures. And I was fascinated as you would look through the microscope and see these little creatures swimming around that you could barely see with the naked eye. And so I started to delve into that more and more and started to become more and more interested in cells, any kind of cell, but especially human cells. And that led to then, well, what about that very first human cell, the one-celled human embryo, the zygote? And was simply fascinated by the fact that you could have this single cell with all the instructions that make a human being in terms of our growth and development, of our metabolism and so on. And it would grow and develop until we had between 10 and 100 trillion cells. So that became my focus then when I went on to work on my PhD, as well as my postdoc and subsequent faculty research.
Absolutely. Well, it really is amazing when you think about, as you said, when I'm talking about pro-life apologetics to people, we talk about how from the moment we come into existence, nothing else has to be added to us. From that, as you said, that single-celled zygote, everything that we need for these really complex beings that we are going to become one day is there from the very beginning. And I love hearing you speak about this because you're very good at taking what is actually a very complex subject and boiling it down to something that most folks can understand. And so we're going to kind of delve into that today, which I'm really, really excited about. That moment when a new human life comes into existence has been said by some to be a matter of religion, or even a mystery that can't be known. I'm trying to think of the exact quote from the Roe decision where it said that wonderful mystery or something like that of when life begins, that we can't possibly know that. But in fact, we do actually know it. It is purely a scientific question. And this paper that we're talking about, this committee opinion from AAPLOG, really delves deeply into this question. I know that I'm asking you to boil a lot down into a relatively short period of time, but can you review for us, what do we know based on scientific evidence about when a new human organism comes into existence?
And you're right, Christina. It's not a matter of religion or belief or mystery. It's frankly dry scientific facts in terms of, as you put it, that moment when a new life begins. And your listeners ought to think about what's the difference between like an egg cell or a sperm cell or even a skin cell and that single-celled human embryo. There's a huge difference. That new organism, and that's the proper term to use for that single cell that's the new human being, does begin at the moment when the egg and the sperm come together. Now, from that point on, it is self-directed. We mentioned how you've already got all of those instructions inside the cell in terms of not just the DNA in our genome, but information stored away in the cytoplasm of the egg that then is activated by that event in time of conception. And that new organism can start to grow and develop. If you look at any other cell type, they will start to grow and then they will split and form two almost identical daughter cells. And that will just continue ad infinitum. There will not be a great difference. If you start with a skin progenitor cell, you're going to end up with a skin cell. But if you have that single-celled zygote, which is an organism, it now will start to not just divide, make more cells, but those cells will begin specialization into, and it is miraculous in some sense when you watch how all of these different tissues and organs form over time, but it's a matter of that being an organism, not just a simple cell. And you know, organisms have different lifespans. I want to bring this up because sometimes people think, well, you've got to pass a certain hurdle to be really called a human being, to be really called an organism. And so we get people where they say, as some of the people on the other side of the debate from us will go, well, not until you're born do you really count as a human being. Do you have any value? Or it's until your heart beats, or until there are brain waves, or a myriad of different obstacles as hurdles that they will put up. But the truth is that that single-celled organism is alive and is a being at that point. It's not just a cell. And some of us go on and grow for years and decades. Some of us have life-limiting conditions that preclude our developing quite as long. But our size and our complexity don't designate that we are a human being.
Absolutely. Absolutely. Well, of course, the law of biogenesis tells us that two human beings can only create another human being. And, I've heard other physicians, I've heard scientists talk about potential human beings. Dr. Prentice, and you've been now studying this for decades. Have you ever come across such a thing as a potential human being before?
There is no such thing.
Yeah.
You either are a human being or you are not. And when a human egg is fertilized by a human sperm, it forms a human being at the single cell stage. The potential is how much development you can undergo.
Yeah.
But the species designation is still the same, whether you're one cell or whether you're 100 trillion cells.
Absolutely. And I don't know if you have the same pet peeve that I do, but I'll use this platform this time again to please implore all of my colleagues to never use the phrase fertilized egg, because there is no such thing as a fertilized egg, right? At the moment of sperm-egg fusion, the egg and the sperm both cease to exist in that moment, and now you have this new human organism.
That's true. It is an embryo, a single cell zygote, and then as we move on, it's an embryo. There's been a term that was tossed out a number of years ago for political use called a pre-embryo.
Mm-hmm.
That also is not a factual term.
Right.
At the point where sperm and egg fuse, that's when you have an embryo. It's a new being, and again, it's an organism physiologically.
That's right. Even if that organism, as you said, has a very short lifespan and doesn't go on to develop very much further, still doesn't negate the fact that that was a human being, a living human being in the time that he or she existed. Even if that was only for a few days, it doesn't negate their humanity.
That's right.
Yeah. This sort of verbal linguistics that is oftentimes used, like saying fertilized egg or pre-embryo, of course, is meant to dehumanize these very young human beings, and there are very real consequences to that dehumanization. Many embryos that are created through the IVF process don't end up being transferred for the potential to then go on to hopefully make it to full-term pregnancy and be delivered. And so then we have this issue now with millions, we think, of embryos that exist and are sort of frozen in time, and many of them end up being destroyed, unfortunately, through research processes.
Right.
Can you review for us not only why is this ethically problematic to be destroying young human beings for the purpose of research, but also has it yielded any beneficial results because of the results of this kind of research?
Well, and to answer the second question, no. There really has been no success in this embryo research. And the one that most people are familiar with is the idea of embryonic stem cells. Mm-hmm. To get embryonic stem cells, you have to let that little human being grow for a few days, and then you literally tear him or her apart to take the cells out of the center of the embryo. Those are the embryonic stem cells. Once put into the dish, theoretically, the story goes that they could continue to grow forever, make any tissue you would want. And I had one U.S. senator tell me they had the potential to cure all known maladies. Maybe a bit of hyperbole. Well, no, complete hyperbole, because after it's been about 27 years now since they first successfully isolated human embryonic stem cells in the lab, started to work with them. We kept hearing, oh, in the next 5 years, we're going to cure all these diseases. Well, in the next 10 years, that story keeps going, and there have still been 0 human beings cured of anything by the use of human embryonic stem cells. There are other proposed things to do for research that, we could study these little embryos and maybe cure infertility. And cure always pops up in usually discussions as to why they want to use these young and most vulnerable human beings. But the bottom line is no good has come from any of that. And maybe that's because of the ethical problem that comes when you have to destroy a young human life, to use them experimentally and instrumentally in the laboratory. As you pointed out, you devalue them. They're no longer a human being. They're simply laboratory material to be used. And, the unethical tends not to result in success.
Yeah, absolutely. Now, and we do have, though, ethical sources of stem cells that have actually been, and you've been very involved in this, that have actually produced amazing results. Can you? I know you have several stories that you can share, but can you just share one maybe avenue of success that we've seen with ethical stem cell treatments?
And we're referring to adult stem cells. There's another version called induced pluripotent stem cells that may also have some potential, but adult stem cells are, think about bone marrow transplants or umbilical cord blood transplants. Those are adult type stem cells. And they have the ability to go in and repair damage in the body. They may do it directly. They may stimulate other cells in a tissue to repair them. Last count, though, there were somewhere over 2 million people who had benefited from adult stem cells. And the favorite story I have, a little girl named Reagan Moore. We feature her on our Stem Cell Research Facts website. She was diagnosed at birth with Krabbe's disease. It's a genetic problem, and usually these little kids undergo neural degeneration and don't live beyond 2 years old. So she was diagnosed at birth with this condition, and they almost immediately did an adult stem cell transplant from umbilical cord blood. Reagan started school a couple of years ago. She's now, I think, around 7 years old. Now, that doesn't mean she's completely cured or perfect, but just ask her dad, and he will say she is perfect. And we've got a lot more of those stories on the website. I encourage people to go see them.
Absolutely, absolutely. Yes, we'll give everybody that website at the end, because it really is amazing to see what can be done with this really innovative research that's being done. And most importantly, that doesn't involve the destruction of an innocent human being in order to accomplish that. And, of course, we know even if there had been good outcomes from embryonic stem cell research, that still wouldn't justify the intentional harming of human beings. Of course, I think one of the most famous examples I can think of that is the Tuskegee experiments that were done on African American men without their consent to see what would happen if we withheld treatment for syphilis. Do we have more medical knowledge now because of those experiments about what happens when syphilis goes untreated? Yes, we do. But it certainly doesn't justify the experimentation that was done on them and allowing them to suffer when they knew that they had a good treatment. So I think it's important to point that out. But as you said, oftentimes too, the unethical doesn't produce good results, and that has been the case with embryonic stem cell research. Perhaps, and you have more information on this than I do, but perhaps in an effort to seem like they're circumventing this ethical problem, in the last few years, scientists in the UK have created what they call human embryonic models, or HEMs, for the purpose of research. Can you describe what these are and what they're being used for, and are these an ethical alternative to embryonic stem cell research?
Well, and they use the term model, embryo model, because they don't want you to consider these to be real embryos. But the bottom line is they are. They meet that definition of an organized organism that is proceeding through normal developmental stages. And it's interesting that in recent years there have been a number of different techniques that have come up besides good old-fashioned fertilization, putting an egg and sperm together, that can produce an embryo. Cloning from years ago, Dolly the sheep, where you transfer a nucleus of one cell into an enucleated egg and stimulate that to start growing. Yep. Who would say Dolly wasn't a sheep? But she started as a sheep embryo through this other type of technique to start an organism. This embryo model idea is to form an embryo, but at a later stage of development. So you don't start with an egg. You don't start with sperm. You don't start with this nuclear transfer. What you do is you take some stem cells, treat it in such a way that they will come together and spontaneously form an embryo in the lab dish. Usually it forms an embryo at what would normally be about 5 to 7 days after conception. People like to think of them as models or like to portray them as models because they want to use them to steady normal development, not just for a few hours or days, but even for weeks and months, and to grow them and manipulate them and so on, which comes back to our problem with the ethics. Is it an embryo? Well, it looks like an embryo, it acts like an embryo. A number of these experiments, including some of those in the UK, have developed human embryo models to the point where you have a beating heart, or they're starting to produce blood cells. There was, in fact, another report just within the last few days of them being able to start develop all the different blood types. Now that normally wouldn't start to happen until a few weeks into normal development. But it's a sign that this organism is actually continuing to move as an embryo. It fits our definition. We're not starting back at the first day, we're starting several days later, but it still functions and looks like, in terms of its structure, like an embryo. So we're back to the whole problem of, should you even be creating these things, especially because the impetus is to use them for experiments, even to grow them and manipulate them, combine them with animal cells to make animal-human hybrids, do gene editing, treat them with toxic substances, and of course then just destroy them at the end of the experiment.
Yeah, absolutely. So these are, even though, as you said, they didn't come into being in the quote-unquote old-fashioned way through, fertilization as we would think of that, they still are human organisms because they have
Yes.
human DNA, completely human DNA. They're human organisms that are alive because they're growing, they're developing along, at least to this point, what we can see, the normal trajectory of what a human being develops. But as you said, I think it's important for people to hear what you said. These are being created with the specific intent to be experimented upon, sometimes in very
Right.
horrific ways. And so this is a real problem. And again, it's just another example, I think, of these sort of verbal gymnastics that people play in order to try and minimize the fact that these are, in fact, human beings that are being experimented upon, even though they're very young and very small. And not visible to the naked eye necessarily, but again, that doesn't change their humanity. And so this is something that I know you are watching very closely. It's something that AAPLOG is watching very closely as well. And we should, all of us, in the scientific community, should be calling for accountability, that this is not how
That's
we
right.
treat young human beings. Moving on to another slightly ethically fraught area here. That question of when human life begins, and is it at that moment that single-celled embryo is created, has relevance when we think about in vitro fertilization as well.
Yeah.
And even though over the last 2+ decades that this has been being done, approximately 12 million children, we think, roughly, have been born as a result of IVF. And of course, each one of those children has infinite worth and value, and how they came into existence doesn't change that. But we know that maybe 90% or more of human embryos that are created in the IVF process never make it to birth, whether they are experimented upon, whether they are destroyed, whether they are just sort of in this frozen limbo state. Can you explain some of the reasons for this? I just sort of briefly went over them, but why
Yeah.
is it that we have so many embryos, so many human beings that never even get a chance really at birth after the IVF process?
And I would point out that we have a chart in the paper that kind of gives a graphical look at all the various places along the way from fertilization to birth. These problems can happen. One of the problems may simply be that we're trying to recreate a uterine or a fallopian type environment in a laboratory dish. And there have even been experiments with mouse and other animal embryos that show that just altering a salt concentration or a particular ion and so on can affect the growth and the health of these little ones still in that dish. So from the point of fertilization, they're in the dish. There are a number that simply don't continue to develop and grow more than maybe a day or two. Doesn't mean they weren't an embryo. As we talked about before, there may have been some life-limiting condition. It may be an external condition of this artificial environment that's causing the problem. After that point, if these little ones grow for 2 to 3 days at least, they're usually graded. They actually will examine them in the laboratory. The embryologist will give them a good look over and decide whether they are, frankly, worthy of continuing to go on in the clinic. Sometimes they'll even do genetic tests, which is another big problem in terms of the ethics. It's really eugenics that's going on at that point. But many of the embryos then are simply discarded. You don't look really that great, despite the fact there have been studies that show that even embryos that don't tend to look all that good under the microscope still can form a normal, healthy child.
Absolutely.
If you survive and you pass inspection, if you will, you might be the one chosen then to be transferred to the womb. And in past years, sometimes up to 6 embryos were transferred. Thankfully, now that's not the case. There tends to be just 1 or at most 2. But what if there are other embryos? Well, that's the point where, as you said, they go into the freezer. And it's interesting that in past years, thawing those embryos tended to be a big problem. Usually only about 50% survived the thawing if they wanted to thaw them out and try to transfer them. They're a little bit better nowadays in terms of percentage, but still many embryos won't survive that. And sometimes they're thawed and simply discarded because there was a decision that the couple didn't need more kids, or they didn't want to pay for the storage fee, or whatever the situation might be. But discarding again becomes an issue here. They may be donated to research in which there's pretty much 100% guarantee they will be destroyed along the way. And so you end up with, as you said, at least 90% or more who do not survive. And I want to illustrate that one more way. You mentioned that roughly 12 million babies that have been born via IVF protocols since Louise Brown, 1978. 90% at least don't make it. That means 108 million little human beings died somewhere along the way. Most of them are not frozen. There's an estimate somewhere around 2 to 3 million in the US in the freezers. But the vast majority don't survive.
Yeah. It's really overwhelming when you think about that. And I also think about, I've cared for many patients who went through the very difficult process of, infertility and treatments and things like that. And so many families, I think, aren't adequately counseled about
Right.
these possibilities, about how many of their children likely won't survive, or, what is their plan if they happen to get 10 embryos that survive. Are they willing to have 10 children, and transfer all of those embryos? So there's all these really difficult things that we really need to be talking about. And I think in large part the pro-life movement and the medical community, really until the last maybe couple of years, a little bit forced by the Alabama Supreme Court decision, really haven't talked enough about. And so I am glad that even though this is a tough issue, it brings up some tough ethical issues, that it is being talked about more. And I really would encourage anyone listening to this, if you haven't spent time really thinking deeply about this, to spend the time thinking about it, because the bottom line is these are human beings, and even at especially at their most vulnerable times, they deserve to have protections. I know there's current discussions going on right now in this country about infertility treatments and IVF, and this is largely an unregulated industry right now in the U.S. and but there are other countries that do it better and put better safeguards in place. So, in your opinion, what are some things that should be done in order to ensure that embryonic human beings' lives are respected and protected in this country, whether it be, when we're thinking about research or infertility treatments or whatever the situation might be? What are some things that we could do, do you think, to
And part of it, Christina, is attitude. We need to realize, again, these are embryonic children. These are human beings at the earliest stage of life. And we need to value those lives, even though they're very tiny, pretty much undeveloped at that point. We need to consider them right alongside any other human being in terms of their value. And especially because when we're talking about IVF, really, the well-being of those children should be paramount importance. We give a number of these recommendations in the paper again, and I'll just hit a few. But one thing that's popped up into notice, even though it's been around for a while, is the idea that before you proceed to IVF, you ought to consider restorative reproductive medicine. IVF doesn't treat infertility. It doesn't get at the root causes of infertility, whereas restorative reproductive medicine does. And there have been several studies that show, even after a number of failures with IVF, couples that then went through this restorative type of procedure were able to have kids. So it's something that really looks at all of the patients involved.
Yeah.
Other things are in terms of numbers. We mentioned the horrific numbers that don't survive. But if you look at the very beginning of IVF, they took one egg, they created one embryo in the dish, that embryo was transferred into the womb, you had one baby born. Yeah. And so not creating lots and lots of embryos just as a hedge or for economic reasons, we might say, because it is a business model that they're using, but considering every individual and their value. Even if multiple embryos are created, not transferring all of these at once, but only transferring one at a time.
Right.
Mentioned freezing and some of the problems with that, and the death sentence that's given to some of these embryos. So we really should consider just not doing freezing of any little human beings. Yeah. The genetic testing that's been done, especially on little human embryos still in the dish, is frankly just eugenic. The whole point there is to to give you a grade and determine whether you have worth to go on and grow and develop versus being tossed or used for research. And finally, when it comes to research itself, I think there ought to be a global moratorium on creating any embryos of any type by any procedure for research, because inevitably every single one of those is destroyed. We need to, again, to value each human life and give it the best protection and nurturing we can.
Yeah, absolutely. I think those are very reasonable things, not in that I know there are a variety of opinions on IVF out there. And I think what's important to point out is that in none of those did you say we should be banning IVF. We're talking about looking at how can we better treat patients, and find the source of their infertility and treat a woman or a man, not just so that they can have a baby, but so that their lifelong health is better.
Yeah.
And then also, how can we respect the lives of these little ones, and how can we make our research more effective? Imagine if we took all of the resources that are being sunk into the creation of these human embryos and then the research on them, which is unethical and destructive. But imagine if we took those resources and instead moved them over to more research with ethical sources of stem cells. Imagine the difference that we could make in people's lives if we were to do that. So, well, Dr. Prentice, I could talk to you for hours, but to wrap up, I know we're at time here. So can you tell us where can people go to learn more about the work that you're doing and the amazing research that's been done with adult stem cells as well?
Let me give you just a couple of things. I mentioned the one website, stemcellresearchfacts.org, where we've documented a number of patients with lots of different conditions and their experiences, life-saving in many cases, with adult stem cells. So I'd really recommend that. And we have the patients pretty much tell their own story instead of dry science from the geek. So that would be a great place to go. The new group that we've started, SALT, the Science Alliance for Life and Technology. Our website is currently under construction, but hopefully within just a few weeks we'll be ready for prime time. And let me give you that website as well. It's salt4life.org, salt4life.org. And we'll have a number of our papers, we'll have a number of blog posts and so on. We'll have podcasts that we've been on, such as this one.
Perfect.
To explain the science. And our mission and our purpose is to help people realize that these are little human beings who have value and worth, and to promote ethical science that does value every human life.
Absolutely. Thank you so much for all of the work that you're doing, and thank you for coming on today to help explain or walk us through some of these ethically tricky issues. So thank you so much for being on. Dr. Prentice is also going to join us in Seattle at the end of February for our national conference, where he's going to be discussing the latest in embryonic biotechnology. So you'll want to make sure that you come so you can hear him. If you'd like to hear him along with many other amazing speakers, please register now for the conference. You can go to conference.applog.org. Remember, AAPLOG dues-paying members, you get a discounted registration, and for non-members, we do have an early bird discount available until November 7th only, so be sure to register today. Thank you to all of our listeners for joining us today. We'll have links to this AAPLOG document we've been discussing in the show notes, as well as a link to our resources page, which is full of very helpful clinical guidance for you in your life-affirming practice. If you find this content helpful, we invite you to support our work by joining today. If you are a medical professional, please join AAPLOG. You can do that at applog.org/join. If you're not in the medical profession, but you care about ensuring that accurate medical information is out there and that pro-life doctors can continue to practice, please join our work by going to applog.org/donate. We can only do this with wonderful supporters like you. Finally, if you liked this episode, be sure to give us a like and a subscribe on YouTube, or if you're listening on a podcast app, please give us a 5-star review. We'll see you on our next episode.